Preclinical data in renal denervation are a wonderful example of how non-clinical animal models and computational models can come together to shed light on an emerging technology in a way that one could never get from a clinical trial. The failure to realize the full potential of a technology like renal denervation in the phase 3 clinical trial was in major part the failure to understand this interplay between application of a technology and understanding of the anatomical and pathophysiologic forces at play. What CBSET did was using an integrated approach which uses sophisticated animal models, complex computational protocols, and then benchtop experiments to fully delineate the extent of the challenge that's confronted in trying to selectively ablate nerves and ganglia outside a specific artery.
Computational modeling is really the ability to predict how devices work. And specifically with regard to renal denervation, the question is, how does the energy distribute in the tissue? And that's something that currently cannot be visualized experimentally, and really you can only assess in retrospect at the end of the experiment. So computational modeling allows you to visualize this and to predict how changes in devices, in device geometry, device settings, will impact the distribution in the tissue.
The obligation we all confront, therefore, is to take the promise of renal denervation, modify our technology, modify our clinical protocols, learn from the non-clinical experiments that CBSET and others have engaged in, and make this the success it really can be.